big project looking at glyphosate degradation. It's a very difficult compound to degrade in soils. And one of the questions was what factors influence it. The two real factors that determine persistence of glyphosate. One of them is pH. The lower the pH, the more persistent it will be, and that's because a lot of the organisms that can degrade it are going to be your bacteria, your lactobacillus and Acidobacter and bacillus species, some pseudomonads and a few of those. But when you drop your pH, you have a big effect then on activity of those organisms that would normally degrade glyphosate. As a synthetic amino acid, it hasn't had time for a lot of mutations and that to take place, so that there aren't a lot of organisms that can degrade it to start with. The second factor, second primary factor for persistence is clay content in your soil. And two factors there. One is the lattice structure of the clay component, so that glyphosate can get entrapped more or less in that clay structure, so it's not readily available for degradation, for microbial degradation. And the other thing, it's also you just don't have the organisms there that can degrade it. So soil pH, clay content, and presence of the organism are the three things that are really important on your clay loam soils and clay soils in Australia. When I was there on a trip before last farming, I believe it was CSIRO was doing a lot of analysis, and that show a 5 or 6 year residual glyphosate on their sandy soils, and could show 20 years of accumulated glyphosate in the clay loam and clay soils. So a big difference with soil type, but in any case, you have a real persistence of this chemical. It's a synthetic amino acid. It's again, there hasn't been a lot of time for a lot of organisms to adapt to utilizing it. The ones that do utilize it are pathogens. So that Fusarium, Phytophthora, and Pythium, and Rhizoctonia, some of those organisms, glyphosate increases the virulence of, and it's also toxic as an antibiotic to organisms that would normally control those pathogens in the soil. Maybe I better stick to your questions.
Look, you're spot on. They're all things that we're really interested to know. So, yeah.
I think we need to remember that in farming, we're managing an ecology.
Yeah.
And our real purpose is to capture the sun's energy and store it as sugar. To do that, it requires 17 elements. For photosynthesis, it requires manganese to split the water. Magnesium is part of that chlorophyll molecule to capture the sun's energy. And then you have all of the other elements that are involved in producing that sugar and storing the sugar and utilizing it. So it's really a multitask process, but one that is really critical to have at least 12 of the essential elements just in that capturing the sun's energy and storing it as sugar, getting it to the right place. And glyphosate impacts pretty much every one of those elements. It's doesn't have much discrimination. It's a very powerful chelator, as well as so that in chelation, it immobilizes an element. In my research, I couldn't find any situation where once that element was chelated with glyphosate, we didn't see any future utilization for it. The length of the flash period for glyphosate is based on how quickly the plant can recover those micronutrients from the soil, not on how quickly the element will be released from the chelation. Once it's chelated, it's essentially chelated for the life of the plant. So it's just a chunk of gravel physiologically in that plant. So very critical to understand the broad scope of this very simple compound. It's a synthetic glycine amino acid. Glycine is the simplest amino acid that we rely on or that we need, and yet serves as a very powerful chelator, a very powerful antibiotic, direct toxicant to some physiologic pathways, systems, and as a synthetic amino acid, it's been shown now that it can actually be incorporated in our own proteins, so that glycine makes up a lot of the protein components. Some of our proteins are about 50% glycine, and glyphosate can substitute for glycine in protein formation. It's thought now that that may be one of the problems that we have with the prion diseases, mad cow and wasting disease, or Kuru and CKJ, and that is that when the glyphosate's there, or when the glyphosate substitutes for copper in the brain cells, that glyphosate has six bonding areas, copper only 2. So if you get glyphosate in there and you substitute manganese for the copper, and that causes the protein folding that gives you a self-replicating protein then that we call a prion for both human and animal diseases. So, but our objective is to capture the sun's energy and store it as sugar. And we need all of those elements functional for us to do that.
To the farmer, so, I very broadly put at the top there, key side effects. From what you're just saying now, to the farmer that looks, they don't realize why the crop might not be productive, but it's due, like with your spectrometer, now you're very quickly able to tell the effect is due to nutrient deficiencies. Because would that be one of the key effects that you would say to the farmers?
Yes, very, very definitely. And we always have had to kind of worry or concern ourselves with hidden hunger.
Hunger, I like that.
We haven't had a lot of tools that we could quickly get that information, and especially with the micronutrients. I know in the Midwest, 60 years ago, we went through and did a lot of soil analysis for micronutrients. We figured we had about 50 years left. And a lot of those soils that we could continue to produce our crops and harvest them and move it off the farm before we really ran out of the micronutrients. Well, we're 10 years past that, or 15 years past that, and just starting to realize we've got a lot of symptoms that we've overlooked. And sometimes with manganese, even with iron, you can have a 10% yield loss or 20% and never see a symptom. And that's because of the inefficiency of the physiologic pathways that those minerals are serving as the cofactor for, or the regulator for those enzymes. And if they're not there, it's kind of like trying to go out and start your tractor without having a key. So we're seeing a lot more recognition now, because the loss of those nutrients or utilization of them and moving them off the farm has occurred over a long period of time. We're just seeing the intensity of that mining the soil, so that we need to pay attention to it. And one of the things that Martha Carlin and her group and Paleobiotica that have biological mixture, a mixture of organisms that can degrade glyphosate. They get about 50% degradation from a soil application in 90 days. And on alfalfa, for instance, even though they still had some glyphosate present, they had a 16% increase in alfalfa yield. And hadn't applied any other fertilizers, but it was in getting rid of that glyphosate that was tying up so much of the mineral composition that it was just as though you had fertilized that field. So very obvious response there that you could attribute to that reduction in glyphosate, residual glyphosate in those soils.